Okay, next, let me start here, as this will become its own separate video that Allison and Jeffrey and others will be able to share if they choose, and we'll be putting this up on the SSURF YouTube channel following the event. So welcome everyone. I'm Dan Powers, Executive Director of the Society for Science at User Research Facilities, and this is another session of our 2021 annual meeting. We are now going to learn about the Facilities Integrating Collaborations for User Science, the Ficus initiative, which was started between the Environmental Molecular Sciences Laboratory, Emsl, and the Joint Genome Institute, JGI, several years ago. It has since grown to include more laboratories, and we're going to hear about that history first from Dr. Allison Hatt, Deputy of User Services at Emsl at the Pacific National Northwest Laboratory, and she's also a SSURF board member. Thank you, Allison. And then we'll hear from Dr. Jeffrey Blanchard, whose climate change research is a powerful example of how a scientist and his team are leveraging the Ficus initiative's value. So let me turn this over to you, Allison, first, and Jeff, you can share your screen as you need to.
Slides here. Let's see how this goes. Okay. So thanks for that intro. And as Dan said, I am just here. Let's see, I need to swap displays. There we go. Is that the right one now you're seeing? Yeah, look okay? Okay, great. You don't see all the presenters.
Muted. Yep, looks good.
Terrific. Okay, so hi everyone. Pleased to be here. I am here just to provide a very quick intro to the SSURF program to set up our next speaker. So as Dan mentioned, Ficus is a partnership between several user facilities. The history, as I understand it, and as a caveat, I've only been at Emsl for about a year, but my understanding of the history, about almost 10 years ago now, Emsl and JGI, which are both funded by BER, started talking to each other and saying, you know, we have these complementary capabilities at our two user facilities. Wouldn't it be great if a user could access those complementary capabilities in a single user project without having to submit separate proposals to each facility, go through those review processes, and kind of align everything themselves? And I think the idea might have actually come from JGI's program manager at the time. But the innovative and hardworking people at Emsl and JGI put in a lot of effort to build this partnership that is both a scientific and an operational partnership, and developed the program as we know it. So the first call for proposals for Ficus Proposals was in 2014. And there is a call now for Ficus Proposals with Emsl and JGI every year. Those proposals are submitted through Emsl's user portal, and they go through the necessary internal reviews at both facilities, and then they are peer reviewed by a panel of subject matter experts identified by the two facilities in collaboration, and the duties of hosting that review panel trade-off between Emsl and JGI each year. And then those decisions, after we get the reviewer results, the decisions on funding are made in collaboration again between those facilities. We then, once a user project is active, we are in monthly communication at least between the two facilities to track the progress of those user projects and make those all important handoffs between the two facilities as a project progresses. So in recent years, the program has expanded to include other facilities. So the National Ecological Observatory Network, NEON, that is an NSF facility. They have participated now in a couple of these Ficus calls, making their resources or some capabilities available. And most recently, this past year, the Biosands beamline at the Center for Structural and Molecular Biology at Oak Ridge was also available through the Emsl JGI call. And we got some great proposals requesting those capabilities. We also now, Emsl has had a couple of Ficus calls with ARM, that's the Atmospheric Radiation Measurements user facility. We're about to do our third one of those. And JGI also has a separate Ficus has a partnership with NERSC and KBase, I believe, with, and has a Ficus proposal call with them as well. So this is a program that continues to evolve and develop. Now that there's a solid kind of operational logistical framework built, it's ripe for continued expansion and evolution. And I think it's a really exciting way to find, working together new ways to bring opportunities to users. And so this program enables ambitious user research projects using multiple facilities with a single proposal, as I said. So looking at the numbers, about 30% of Ficus proposals for those JGI Emsl calls come from PIs who are new to the facilities. And that's an important metric to us. It's really, that's a higher percentage. I think our standard calls otherwise at Emsl, at least, it's like maybe 20% are new. So we are reaching more new researchers, and that's one of our goals as a user facility. Those Emsl Ficus calls have now funded 96 projects since it started in 2014. And we have record of 61 publications resulting from those and counting. And 23% of those publications appear in what we think of as the premier journals, PNAS, Nature series, or Science. So just in terms of the standard impact metrics, kind of publication-centric metrics, this is a really impactful, successful program. And our next speaker will talk about kind of the researchers' experience of that success. Finally, then I just want to mention our next calls for Ficus proposals will open in December and January. So you can learn more at our website, emsl.pnnl.gov, or over at the JGI website. They have a lot of great information. Eventually, though, you'd be coming over to the Emsl website to actually submit your proposal. If you can't find the information you need, please feel free to reach out to me or to other partnering facilities, and we're here to help. So with that, I am going to pass it over to you, Jeff.
Thanks, Allison.
Thanks, Allison, for that introduction, and to Dan for inviting me to give this talk on my research experiences working among user facilities. And thanks, Eli, for a great opening talk. We're starting to move around many tens of gigabases that are say gigabytes. We go back and forth between bases and bytes in the DNA world. And that's one big challenge for us, is moving it through different analysis tools doing that. So today I'm going to talk about my first Ficus project, which is integrated genetic data from the Joint Genome Institute and mass spectrometry data from Emsl, and some of the new science that comes out of it. Give a little bit of history and the people involved in this. So I try not to put too much of the details of the science, but still have some of the science results that are coming out of it. So a lot of my work in this area started meeting Jerry Melillo, who's a climate scientist, ecologist, who started some experiments at Harvard Forest. And about 10 years ago, I did a fellowship there with funding from Harvard University to start doing genetic work at Harvard Forest, and met Jerry. And to me, the sort of synergies in what I do and what he has set up were pretty obvious. So one of the things that Jerry has set up and got funding for is an NSF long-term ecological research site, which also could be considered a facility in that there's a lot of users of this site, including the experimental warming plots that Jerry set up. So there are cables going 10 centimeters underneath the forest floor that heat the soil all year round, 5 degrees above the control temperatures. So in the summertime, when the soil is 20 degrees, the warmed plot is 25. In the winter, when it gets down to a few degrees, like 2, the experimental's at 7. And so here's one view of the plots, the patchwork of plots, when there's sort of a light layer of snow, and so that it's all melted off the heated cables. And so my first project with Jerry was actually a Joint Genome Institute Community Science Program Award, working with Jerry and several other scientists. And basically, we're asking questions about microbial community dynamics across actually 3 separate warming experiments that him and Sarita Frey have set up, looking at how the community has changed. And I'm just going to give a quick synopsis of some of the results. One of the cool things, if we look at the DNA and RNA, so we just take soil and we extract the DNA right from that. And from that, we get a beautiful view of the organisms that are in the soil just from the DNA and RNA. We can see viruses, double and single-stranded ones, different types of bacteria, slime molds, fungi, and my favorite, the tardigrades. So it's just this wonderful view of this food web just by looking at DNA or RNA in the soil. And one of the things that we saw were significant differences, particularly in eukaryotes dominated by the fungi, have gone down in abundance and expression of their genes with long-term warming. We've also seen changes in viruses in those plots. If we look at the genetic data, we can see just a multitude of different pathways. Some of those related to biogeochemical cycles, carbon cycle, nitrogen cycle, sulfur, iron, and phosphorus. And we can see pathways that have either been higher in the control or stayed higher, or those that have been elevated in the heated plots, for example, utilizing chitin, which is a part of the fungal and insect cell walls. We can also see changes in sulfur, iron, and phosphorus cycles. And these are elements that are not traditionally measured easily by ecologists, but we can get a perspective on them from looking at the genes that they're expressing. And so this sort of gene-centric view, which is sort of part of who I am, I also realized that we're missing the molecules and what's going on in the abundance of those molecules. This is sort of an old classic paper of making a diagram of metabolism from genome sequence data. And I just want to highlight on the left-hand side, you can see different sulfur and nitrogen cycle-related elements being taken up. The top, there's iron, the bottom are different carbohydrates. And so cells, and there are thousands of different species of bacteria and other microbes in the soil, are constantly taking up, expelling, dying, releasing these molecules. And so wouldn't it be wonderful to have a view of those molecules to help us integrate how these microorganisms might be interacting with these biogeochemical cycles. And I think I gave a talk at one of the DOE meetings, it was probably like 2016, and I met Mary Lipton there, and she told me about the Ficus, both Emsl's user capabilities as well as the Ficus program. And so for me, it was like this great opportunity to try and integrate the genetic data with a lot of this molecular data, particularly the soil organic matter, the metabolites that are present, the lipids, as well as looking at the proteins. So I put together my first successful Ficus Award, working with some of those same group of collaborators, but new ones, including Marcus Colliwaite, who had specific expertise in working with the FTICR-MS data for our soil organics. And so this has been a wonderful project. We've already got 3 papers with probably at least 3 more coming out of it. And a lot of it was driven by 2 really incredible graduate students, Lauren Altillo and William Rodriguez, and a couple of undergraduates in the lab who actually worked on the data for 2 years and did some incredible work. But that group and sort of the other group of associated scientists really wouldn't have gotten anywhere without the interactions from people at JGI and Emsl. It really took my ideas and elevated it in different ways into directions that I never really thought of before and that I'm pursuing right now. So I'll give you a few examples of that related to it. So the first part of what we did with JGI was we actually used and developed with them a novel procedure for doing the soil metagenomics. And that if you're working just with the soil itself, it's really difficult to assemble the DNA back into genomes that represent the organisms. So we did sort of a reduction in complexity approach by using some centrifugation, filtration, and then sorting individual cells into pools of 100. So again, 100 rather than doing one single cell genome for reasons of increased efficiency, and then reassembled the genomes from those 100 sorted cells. And so the quick upshot of this is these CSP genomes, these cell sorted genomes, we could get many more genomes together and assembled, about an magnitude order more than using the traditional metagenomics approach. And this is a big step forward. And not only did we get more, but we got lower contamination in our genome bins. One of the things that we saw, and I guess we knew we would see this, is these black dots in these outer circle represent novel species of bacteria. And as you go in family, order, class, domain, phylum. So one of the genomes we assembled is a potential new phylum bacteria. Nearly over half of them are new genre of bacteria. So a lot of new diversity out there to be discovered. It's one of the things I love about working with the DOE user facilities is, you can have hypothesis-driven research, but you can have discovery-driven research. And that's very hard to fund by other mechanisms. And one just huge example of that is we didn't anticipate when we were doing our cell sorting, we gated it for bacteria, but there are viruses that are bigger than some bacteria. And these are often called giant viruses. And we discovered new giant viruses, 16 new lineages, including what's still the world's second largest viral genome, just from soil here in Massachusetts. We think there's other bigger genomes out there. This never would have happened without the insight of Frederick Schultz at the Joint Genome Institute to even look for them, because sort of the typical algorithms we use in processing the data sort of focus on bacteria. Also, as part of the Ficus Award, sort of more on the Emsl side, Marco Kaiwait's lab, working with a group in Switzerland, show that there's a significant decrease in electron accepting and donating capacities to the soil, particularly in the mineral layer. And a lot of that's driven by changes in the phenolics and lipids in the system. William Rodriguez in my lab, comparing the heated and controls, showed that there were more than 100 more unique molecules in the control sample than the heated, sort of leading to the hypothesis that many of these molecules have been used up, potentially carbon-related ones, by the microbes in the soil, so that the heated soil is now relatively deplete in molecular complexity relative to the controls. That just gave me a new idea of there's ways that I look at and measure genetic complexity, and I've not thought of applying those to molecular complexity to show that change. And then if we look at sort of comparing the Emsl metabolomics data, most of the molecules that we looked at went down in the heated plot relative to control, and it's likely a result of a decrease in fungal and microbial biomass in general. But there were a couple compounds. One is sulfurose, which actually is an inducer of cellulase and might be responsible for the breakdown of more complex carbohydrates in the heated plot. And the other is maltotriose. And if we look over at our gene expression data from JGI, we can see that there's also a significant increase in maltose and maltodextrin related genes. William took both the enzyme data, the big E's here, which are from JGI, and the compound data from Emsl, and looked at those that had interactions with each other through a database of metabolism called KEGG. And the edge of this depict warming effects, and the clusters are based on some common metabolic pathway. And so doing this joint analysis, he could pull out clusters of compound enzymes that related to antimicrobial peptides, defense mechanisms in plants, spore-related compounds, enzymes involved breakdown of plant cell wall materials. So that's just some of the things that have come out of working on our first Ficus Award with JGI and Emsl. Again, a lot of this, oh, I forgot to mention a key point in that earlier slide about Lauren and William. Right after I got this award, we immediately applied for DOE graduate research awards. And so Lauren spent a year working at JGI and helping develop the methods, and William spent 5 months at Emsl working mostly with their computational biologists in the analysis of the data. Again, that was huge in making these type of interactions and these insights possible. So this, about 2 years ago, just before the pandemic, I got funding for a NSF Long-Term Ecological Synthesis Award. So these are working across the various LTER sites that NSF has established. Many of them, like Harvard Forest, are like 30 years old and been collecting data. And we're testing specific hypotheses related to climate change. One of the aims include the development of a reference database for ecological genomes and response traits. And soon after this group was formed, one of the group members, George Rodriguez, pointed out that there is a new Ficus call that not only integrated JGI and Emsl, but the NEON facilities. And so NEON is the National Ecological Observatory Network. It's funded primarily by NSF, and they have sites established around the country, and they have archival soil samples to go along with their data. And so we kind of the expertise, and we had sort of the interest and questions to write a proposal that utilized the NEON soil data using JGI genetic and Emsl mass spectroscopy capabilities similar to our prior award. And so we're getting that up and going and are really excited by that. So I kind of want to give in the last few minutes here a couple little sort of almost promotional talks for the user facility, to the user facility people, but from my perspective as a user. So in looking at one of the reasons why I really enjoy working with these DOE user facilities and the NF as well, is not just the technology, many of which are not readily available at my university, but really it's the people and expertise, and it's my ability to interact with them that's sort of led to new ideas, new analysis pipelines and methods. It enables me to do discovery science in a way that is really not possible through traditional grant mechanisms. So of course, important preliminary data for those proposals. We can sort of leverage it in many different ways. It's also creates data that's really hard to fund in another project. You know, it's a little bit of chicken and the egg for some people. Do they get the sort of more traditional grant that funds work and the people? But many of the projects that we do, both through JGI and Ficus, would be hard to include in a traditional NSF and a sort of a DOE academic side proposal. And these projects have provided funding for graduate students and classes as well. So it's really amazing for me. The project management that's associated with them is really great. The textbook expertise is amazing. I've had sort of graduate students and postdocs do similar things, and my experience is the quality has always been better working at JGI and Emsl, and of course they have professional staff. And so while I could do some of these things, these user facilities that I work with do much better, and they allow me to put my resources elsewhere. So I'd really like to thank some of the people in my lab, but really the staff and scientists at JGI and Emsl have been terrific working with, and I'm already enjoying working with the folks at NEON as well. I do look forward. We have a lot of computing challenges, and I was listening sort of eagerly to Eli's talk, and look forward to other solicitations involving these computing facilities. Thank you. Happy to take questions. Feel free to contact me at that email that was just listed there, jlb@umass.edu.
All right. Thank you very much, Jeff. Great detail, and thanks for prompting or otherwise describing why it's been helpful and efficient working with the Ficus network. Hopefully it's gotten some people who are in the audience thinking of ways to connect in with, or even perhaps how their facility could become part of a Ficus network. I wonder if I could ask that clarifying question, Allison. Is Ficus open for additional partners? Or stepping back at 10,000 feet here, what other kinds of combinations of facilities are possible?
It's an exciting question, and that's one that we're all thinking about. I know Emsl has been in conversation with many other facilities as potential partners. I believe NSLS participated, NSLS2 participated in a call as well a few years back, and I failed to mention that. But yet we are in conversation with some of the synchrotrons and other facilities, and are kind of actively working on some new, adding some new capabilities to Ficus. And I think, in theory, the sky's the limit as to what, how much we could partner, who's working with whom. I think it's a really interesting question to imagine what happens, can this program take off beyond Emsl and JGI? So far, those two facilities are at the center of it. But could a nanocenter and a synchrotron kind of under the Ficus umbrella, using some of that same mechanisms that have been developed, have their own Ficus call? So yeah, I think those are exciting questions, and I certainly don't have answers to them.
Yeah, and I'll add that there's no way that I would have submitted separate proposals. I would have looked at it and went, this is sort of too complicated. You never know. You got two different review panels that aren't even seeing the separate each other. Mm-hmm. And to me, it would have been too much of a risk, I think, in terms of my time to do that. And so having those two under the same umbrella, and the same with adding NEON, I don't know that I would have submitted this NEON proposal if that hadn't already been part of this. So that made a big difference for me.
Nice. A quick comment has come in here from Alicia, who presented earlier, saying KBase would love to work with Ficus to streamline and support the data analyses from these proposals as well. So perhaps there'll be a follow-up conversation that can be had between the three of you, or Allison, if you take the lead on that.
Yeah, it's great. We actually are working with KBase now on the analysis of some data. Actually, it's not from our JGI project, but from some internal sequencing data that we have that we're using as part of it. Part of it, JGI has a really nice pipeline, and so they provide a lot of the computation that we need unless we want to go back and redo it in different ways. But yeah, that's a really great way to interact, and yeah, I'll leave it at that.
All right, I see a couple other comments here. Shane Cannon is saying that NERSC continues to be interested in combining experimental and computational resources. So if there's a NERSC conversation to be had, Shane's going to be speaking in the next panel, I recall. So you can hear a little bit more about what NERSC is doing in terms of remote access to experimentation and facilities. Also, Bill Miller had asked, how did the Ficus engagement with NEON go, and are there particular benefits and challenges working with non-DOE facilities?
Mm-hmm. So I was not involved when NEON was first brought into Ficus, so I can't give you great perspective there. There are challenges working with, I would expect, a non-DOE facility, or even within DOE, working with facilities that are funded by, say, BES instead of BER. We have some differences in what we're looking for in proposals. For example, in BER, mission alignment is a really important part of the review process and review criteria. BES has a different approach, and so kind of determining how we reviewing proposals could be a little challenging there. I think with NEON, it worked rather well because they're involved, they're providing samples, but they're not like providing equipment, or I think putting a lot of funding resources in. So I think that one went pretty smoothly, but yeah, I expect as we go beyond DOE, there would be some additional challenges, perhaps unforeseen.
Yeah. And from my perspective, I had to get a separate letter from NEON, which was really easy. You know, basically, these are the samples that I'm requesting, making sure there's enough material available in those samples for the experiments being done at Emsl and JGI. So there's some back and forth and coordination that I needed to do ahead of time, but that was really, that was a piece of cake, actually. And so I thought it was all very seamless at my end.
Thanks. So a very positive overview of Ficus in general, and then the details of your particular research too, Jeff. Very interesting. It seems that there might be some other conversations that have gotten prompted by your presentation that can happen online and, excuse me, happen offline. And I look forward to helping to make those connections with everyone. Okay. Our next session will just be starting in about another 5 minutes. So thank you for taking the time this afternoon for a half hour, and we'll see you in the next session.
Okay.
Great, thank you.